Okay, can you see the screen?
Yeah.
Okay, cool. All right, so as it says on the tin, I'm going to talk to you all about brain aging biochemistry and therapeutics, new approaches to slowing the process of aging in the brain. And I gave a talk previously on geroscience and venture capital to the Aging and Longevity Society. Feel free to go back onto YouTube and watch that as a precursor to this. So these are my disclosures. These are companies that I either consult for or have substantial equity holdings in. And this is the outline. By the way, this picture here, this painting, is The Fountain of Youth by Louis Cranach the Elder. And you can see old, infirm people on the left, disrobing, and then getting into the Fountain of Youth, and then coming out young and rejuvenated on the other side. So that's ideally what we can develop one day with drugs. So I'll talk to you about brain aging itself, new approaches to brain aging therapeutics with a focus on Alzheimer's, and we'll have some time for Q&A at the end. So the brain is the most complex structure in the known universe, and aging is one of the last great mysteries of biomedicine. And so suffice to say, in this 45-minute or so talk, we're not going to be able to crack that riddle, but I will hope to give you, to sort of whet your appetite, pique your interest in a number of topics in the field as a sort of brief rapid fire survey, and then you can dive deeper into those subjects on your own time. If you're interested in aging, the biology of aging specifically, you can go back to a prerequisite lecture, another lecture, this is the third, also given at Oxford, on geroscience, the biology of aging, in which I get into some of the underlying molecular mechanisms of aging. So those mechanisms are sometimes called the hallmarks of aging, or the 7 deadly SENS, as our mutual friend Aubrey de Grey coined, that actually predated the hallmarks of aging. And they break down into these categories. And those are obviously driving aging in the brain, but there are some additional hallmarks, maybe a level up, a layer up in abstraction, that apply specifically to the brain. So these are hallmarks specific to brain aging that I and others have put together. And of course, this is not an exhaustive list, but here are some of them. Neuroinflammation is a big one. This results in and is driven by impaired phagocytosis and activation of glial cells. We'll get into that process. Failure of bioenergetics. So particularly insulin resistance in the central nervous system, also called type 3 diabetes. Excitotoxicity, reactive oxygen species, and calcium mishandling. Mini strokes, which result in reduced blood flow and vascularization. Loss of anti-apoptotic trophic support. So you may have heard of neurotrophins like BDNF and NGF. These are critical signals that basically tell neurons to stay alive. Neurons, if they don't receive these pro-survival factors, they automatically apoptose. And finally, blood-brain barrier leakiness. So we have specialized cells that keep the bad stuff out of the brain, such as pathogens and such as large molecules that should not be entering the brain. And that becomes leaky with age and with certain diseases. So unfortunately, cognitive function declines with age. You can see here, the blue-green lines are actually raw horsepower, fluid, or working memory tasks. These decline starting in your 20s. And then, as some consolation prize, we have the accumulation of knowledge, general awareness about the world that rises as a function of age. So that comes as no surprise. So we may gain wisdom, but we actually lose IQ points as we age, as you can see in the red fluid intelligence chart. And it's often said anecdotally by the famous mathematician, for example, G.H. Hardy, who came up with Hardy-Weinberg equilibrium, the biologist will know. He said, no mathematician should ever allow himself to forget that mathematics, more than any other art or science, is a young man's game. At that time, it was mostly men. Now 30% of math PhDs are women. But it's often said that, by the time someone is 30 or 35 in math, they're basically washed up. That's probably because mathematics is one of the most G-loaded or cognitively demanding disciplines out there. There's a really good movie, by the way, about the work between G.H. Hardy and Ramanujan, and it's described in this film, The Man Who Knew Infinity. So the bottom line is, while you're young and your brain is working at peak capacity, don't squander that opportunity. Don't waste it by taking empty but prestigious box checking type jobs just for the money. You know what kind of jobs I'm talking about. Maybe those are those jobs in the city that you're not so excited about. So I've told you some bad news. The brain shrinks about 1% per year after age 30. This is data from the National Institute on Aging. But there's good news as well, they find. There are cognitive superagers. There's very widespread in the data here, the pace of decline. And so you can actually preserve cognitive function quite well. But the question is, how do we go about doing that? As a fun fact, the brain is one of the most delicate, evolutionarily new and high performance organs. It consumes 20% of ATP, but 5% is 5% of the mass of the body. So it's very energy hungry. And therefore, it's very sensitive to aging and health. It is the canary in the coal mine. So when your health declines, or maybe you haven't slept well or whatever, you will first notice it in a decrement in cognitive function. Although sometimes you don't notice it if you are too cognitively impaired, because one of the unfortunate ironies is as we become less cognitively perceptive and aware, we also lose awareness of our own cognitive capacity. So it's a subtle decline. So Alzheimer's and other forms of brain disease kind of sneak up on you as a result. So what actually happens to the brain with age? Well, on the tissue level, it shrinks. They say if you don't use it, you lose it. Well, even with the brain, even if you do use it, you will lose it. At least most of us will. It's just a question of delaying that inevitable progress. So that shrinkage in volume, both gray matter and white matter, occurs about 5% per decade, starting at age 40. Particularly in the dorsolateral frontal cortex, area 43, the hippocampus and the temporal lobe, and the striatum. In normal aging, the shrinkage is due to synapse loss, cell shrinkage, as well as demyelination. It is not due to apoptosis, and this was a big controversy in the field. But in late stage diseases such as Alzheimer's, atrophy is due to apoptosis and the actual loss of cells. So you can see here, even in mild cognitive impairment on these MRIs, you can have, so here's the healthy control. You can see the ventricles are relatively small. There isn't that much volume loss. Whereas in various forms of mild cognitive impairment and AD, you get the enlargement of the ventricles, you get reduction in or expansion in the sulcus and gyrus size, and so on. So even with relatively subtle declines in cognitive performance, we actually get large changes in tissue atrophy. And so it's not usually cells disappearing, it's actually the loss of synapses, the connectivity of those cells. And so you can see on the dendritic spines, as pictured here on this cortical neuron, there are dendritic spines that stick off of the dendrite, and this is where the synapse is actually formed. And one neuron can connect with thousands of other neurons. So the loss of those connections is actually what's driving normal cognitive aging, it seems. So you can see in this super resolution image a normal neuron and then an aged neuron that has lost its dendritic spines. Dendritic spines come in multiple flavors: Philopodia, thin, stubby, mushroom, cup-shaped. It's a spectrum to do with a degree of connectivity. And you can see the pre and postsynaptic synapse here between two neurons, and you can see the postsynaptic density that contains a lot of receptors that are just sitting waiting for the presynaptic neuron to release vesicles containing those neurotransmitters and neuropeptides. So the shape of the spines corresponds with the level of maturity of the spine, the connectivity. It's sometimes called Hebbian learning, neurons that fire together, wire together in synchrony and at certain frequencies of firing. And so the strength makes them look more like mushrooms. So the research that showed that synapse loss and loss of dendritic spines is causative driver of normal cognitive aging comes in part out of the lab of John H. Morrison at that time at Mount Sinai, who was actually a mentor of mine. I worked in his lab, and he was recently inducted into the National Academy of Medicine, and it's well-deserved. And what he and others have showed is that with normal aging, neurons are not dying, but the synapses are being lost. And white matter lesions occur, and there are fewer spines and dendritic arborization. So the number of dendritic arbors or dendrites coming out of these neurons declines. And you can see that here with the young compared to the old neurons. So why are these synapses being lost, and why ultimately are whole neurons lost with age? Well, there are a lot of reasons. We'll get into some of them, but one common theme is proteostasis failure. So the inability of the neuron to turn over its protein components. And neurons have a lot of receptors. They're constantly turning over these sort of signaling proteins. And so they're highly reliant on proteostasis. They're also very long-lived cell types. So if you have a cortical neuron, it has to invest in its own longevity. By the time you are, 10 years old, you already have most of the neurons you will have in life. It's just a matter of fine-tuning the connections. And so we cannot afford to lose neurons. So neurons, they invest in their own longevity. They're almost like the supercentenarians of cell types. And that requires them to invest a lot in proteostasis. And part of that is because neurons cannot do what a lot of other cells can do that have the option of doing, which is basically calling it quits, throwing in the towel, and undergoing apoptosis, a programmed cell death. In your liver or your pancreas, if that cell undergoes a large amount of stress, like ER stress, it has the option of saying, okay, this is too much for me, I'm just going to apoptose and get replaced by a progenitor or stem cell in proximity. Neurons can't do that. We do have neural stem cells, but this is a very controversial area, but in adult developed humans, it looks like those neural stem cells are not really doing that much, unfortunately. Anyway, so this process of proteostasis, you have various types of proteostatic stress. It unfolds proteins or causes other types of post-translational modifications to those proteins. They can be handled in a number of ways. The default way is they just aggregate and gum up the works and cause aging. An alternative is chaperone-mediated folding. We have these little Pac-Man-like proteins that fold misfolded proteins, and heat shock factor regulates a whole lot of these. The proteasome can actually degrade relatively small proteins that have been ubiquitinated. There are efforts underway to activate the proteasome, as there are efforts underway to activate chaperone-mediated folding. But in my view, and of course I'm biased, I think autophagy is the most important proteostatic mechanism in neurons in particular, and many other tissue types. And in autophagy, and we'll get into this, you have various subtypes. One is called chaperone-mediated autophagy, or CMA, which can break down certain proteins, maybe about 40% of the proteome, whereas macroautophagy can break down pretty much everything, it seems, and not just proteins and protein aggregates, and even properly folded proteins, but also whole organelles. So autophagy can break down mitochondria, then it's called mitochondria. It can break down ribosomes, which are huge complexes. It's called ribophagy. It can break down microbes entering the cell, and then it's called xenophagy. Basically, any structure in the cell, including the nuclear envelope, can be broken down by autophagy, and there's a flavor of autophagy for whatever organelle that you want. So this is the basic process of autophagy. You have the formation of this double layered membrane, the isolation membrane, or the omega zone, usually budding off of the endoplasmic reticulum. And it encapsulates, with the help of certain cargo loading adapter proteins, whatever it is you need to degrade. Let's say it's mitochondria in this case. It forms the autophagosome. The autophagosome itself cannot do the job of degradation. It must fuse with the lysosome. The lysosome is kind of like the garbage recycling heap of the cell. It contains hydrolases and a lot of acidity that fuses with the autophagosome and breaks down its components and recycles those macromolecules, whether they're carbohydrates or lipids or proteins, you name it, into amino acids for reuse in the cell. And so the lysosome for many years was kind of ignored as kind of an unglamorous function within the cell. But all of the roads keep coming back to Rome of, in aging at least, especially brain aging, of lysosomal function. And we're finding that in the brains of patients with many different neurological diseases, there is a failure at the level of the lysosome, and particularly the acidification of the lysosome. It can't get acidic enough, kind of like your stomach acid. If your stomach is not acidic enough, the enzymes, the proteases cannot work to degrade your meal. Just the same with a lysosome, it gets clogged up with junk as a function of age and disease. So why do I say that autophagy is so important for the brain and neurons? Well, it is the most sensitive tissue to autophagy loss. So if you genetically ablate the ability of cells selectively in one cell type to undergo apoptosis or undergo autophagy, the result is neurodegeneration. But if you do that also in the whole body, you have a conditional adult knockout of autophagy or knockdown of autophagy in the whole body of an animal like a mouse, you would take your guess, you would take your pick, which tissue would break down first, which is the most sensitive. And it turns out that it's actually the brain. We find pathology first emerging in the brain. Even though autophagy is knocked out in the whole body, the brain is the most sensitive. So that's quite a clue. It's also quite interesting that the mice die additionally from bacterial infection. So that's another clue that autophagy is critical for the innate immune system, and we can get into that. So knocking out autophagy during development is lethal because autophagy is so important for every process, but conditional adult knockout results in neurodegeneration. Suggests neurodegeneration in humans is to do with proteostasis failure or whatever it is that autophagy is doing. Autophagy does a lot with metabolism as well, the availability of energy. And it's the first line of defense against intracellular pathogens, pathogens that get within cells, which is, by the way, another driver of neuroinflammation, is brain infiltration by microbes. So, of course, it sounds like a good idea to enhance autophagy pharmacologically. There are some molecules that do this. Rapamycin is known to do it. Metformin does it very weakly. But what if we had a mechanism that was not mTOR dependent? Because that comes with some side effects. And indeed, a number of groups have set off to do this. And one of the catalyzing factors was actually the Nobel Prize being awarded in 2016 to Dr. Osumi in Japan for using yeast genetics to tease apart the underlying mechanisms, the molecular genetics of autophagy. So in brief, when cells age and become old, they accumulate junk of all types, and they rely on autophagy primarily to clear that junk. Also, fasting and exercise induce autophagy. Those are some of the best known interventions for healthy lifespan extension. And it turns out that, at least in mice and other organisms, they're necessary for the lifespan extension and much of the health benefit from fasting and exercise. So if you do not have intact autophagy, you do not get the benefits, or as many of the benefits, from fasting and exercise. And indeed, not only is autophagy necessary for lifespan extension, it is also sufficient for lifespan extension. So a number of groups, including Beth Levine, who sadly passed recently, a great contributor to the autophagy space, particularly in the oncology field, her group had shown that you get about a 17% lifespan extension, median lifespan extension in mice when you increase autophagy. And that is not even an optimal level of increase. We don't even know if we turned up the volume to the right amount. It could be too little, too much, who knows? So anyway, that is a very promising result because that's a pretty large effect size in wild type mice. So as a shameless plug, there is indeed a company in Oxford, primarily based in Oxford, developing drugs for autophagy enhancement. It was founded in 2019 by yours truly, working with Apollo Ventures. And we've raised $5 million seed round thus far, and we're raising a $10 million Series A round soon. If you or one of your wealthy biotech VC friends wishes to invest, you can get in touch with me. And we have quite a strong team assembled. So Jens Eckstein, who ran GlaxoSmithKline's venture fund, and Peter Hamley, who is an experienced drug hunter, medicinal chemist from pharma, as well as Guido Kromer and Frank Matteo, Both of these gentlemen I recruited to the company because they are some of the most prominent voices in the geroscience field. And indeed, Guido Kromer is one of the authors on that seminal paper that's been cited about 4,000 times now, The Hallmarks of Aging. So we're very pleased to have this sort of dream team assembled. And to my great pleasure, it seems to be going quite well thus far. We also have a couple other faces you might recognize from the Oxford ecosystem, our rock star scientists in Oxford, as well as some spread all over Europe. I would invite you to check out some of our publications and some of the news stories. And one of them that I'm proud of is we actually won Best Pitch competition at the Financial Times Pharma Conference a couple of years ago. So anyway, do check out Samsara, especially if you're looking for a job as a biochemist or molecular biologist in Oxford working on autophagy. There are a couple of other companies working on autophagy enhancement. Samsara was one of the first, but a couple more have come out of stealth mode since. And they're all working on slightly different angles. Some of them are a little bit over-concentrated, I think, on one target, the trypamyl ion channel on the lysosome. But there was a recent piece of positive news where Merck actually acquired one of these companies, San Diego-based Calporta, for about 500 million in biobucks. I should note that biobucks does not mean cash up front. Biobucks means some amount of cash up front usually, and then earnouts or milestones, so clinical trial success milestones, as well as royalties on net sales. So you see these eye-popping deal numbers in pharma and biotech, but don't be misled. It's usually the cash up front trading hands is usually substantially less than the biobucks headline number. Anyway, enough about autophagy. Let's talk about the immune system. So some of these slides came from a previous neurology conference that was really interested in the neuroimmune interface. I won't say that this is a particular area of deep expertise for me, but one that I think is very fascinating and kind of underappreciated. So hopefully there's some immunologists in the audience who will appreciate it as well. So I mentioned microglia. Microglia are the brain's resident macrophages. They clear out the junk. They function as the immune system in the brain. And you can see when they get activated, they actually show this sort of lightning bolt-like phenotype. And that activation is actually a sign of chronic neuroinflammation. That's not so good. And when there's a lot of inflammation, the glia become a little bit enthusiastic and they mediate synaptic pruning. So synaptic pruning is essential for forgetting and for neurodevelopment. But if you overdo it, you actually lose memory and cognitive function. So it's this sort of finely tuned balance, and the microglia can kind of go wild and prune too much. So they're actually originate in the bone marrow during development. They become microglial progenitors, migrate to the embryonic brain. They become self-renewing, mature microglia, where they then mediate various functions. So they're involved in neural development with the growth factor production, tissue surveillance, phagocytosis, getting rid of apoptotic bodies and other debris. They support other neuroglial cells, such as oligodendrocytes, which insulate neurons. They kind of create the myelin sheath, which enables conduction of signals to occur faster. But most importantly for our interests in memory and learning, they actually mediate synaptic pruning in the process of forgetting. So that the major immune cells in the brain, they're 10% of all the brain cells. They monitor extracellular calcium changes as a sign of danger. They sense PAMPs and DAMPs, pathogen and damage associated molecular patterns. And they become activated when the blood-brain barrier fails. And you can see in this traumatic brain injury model, this stain of activated microglia. So we actually really need microglia, whereas if you acutely ablate them, you actually get neurodegenerative symptoms. So in the ablation, the red line here, the animals get acute ataxia with a pharmacological ablation, and also are critical in multiple sclerosis model as shown by this group. And they also fail to function on the rotorod, their sort of coordination, physical coordination abilities. So we really need microglia. You can't just get rid of them. But they do mediate neuroinflammation. So like everything in biology, it seems, they are a double-edged sword. And so when microglia and astrocytes get together to sort of overreact to inflammatory stimuli, they cause neural death and also synaptic pruning. So what you want to happen is that inflammation stops cognitive function and enables time for the microglia and the astrocytes to try to solve the problem, whatever it is. But if they cannot solve the problem, then that's when things go haywire and neurons die off. And I encourage you to look into this mechanism of action further. So what causes inflammation writ large? Well, there are a lot of things. Here's a list. One is immune senescence. So immune cells, we know, decline in function with age. That enables microbes to run rampant. Tissue damage, such as chronic unhealed injuries, cause chronic low-grade inflammation. Sugar and metabolic syndrome. There are a lot of mechanisms behind this, but one simple one is the formation of advanced glycation end products. So proteins get glycosylated, and this results in those proteins binding the RAGE receptor for AGEs, which enhances inflammation. Gut barrier permeability is a big one. We'll talk about this, also known as leaky gut. Microbes and PAMPs enter the local gut stroma. That causes local inflammation, and then if it's bad enough, chronic systemic inflammation. Another one is DNA damage and the de-repression of transposons. These are endogenous retroviruses that actually make up a huge chunk of our, the dark matter of our genome. Cellular senescence, this is a big one. You can go to one of my prior lectures to learn a little bit more about that. And this pro-inflammatory cytokine milieu called the SASP, senescence-associated secretory phenotype, that is responsible for sending out this pro-inflammatory signal to the immune system. It's a come eat me signal to the immune system. So senescent cells have undergone stress such as DNA damage, mitochondrial failure, et cetera, or telomere attrition, and they need to be cleared. This is a mechanism to prevent the occurrence of cancer. And it sends out this pro-inflammatory signal to the immune system to come eliminate the senescent cells. If the immune system is too aged or compromised in some way to get rid of the senescent cells, then you get this buildup of pro-inflammatory load. And so that's when the senolytic drugs come in. At Apollo, we have a portfolio company working on this, and there are a handful of others out there, developing drugs that will selectively ablate or eliminate these senescent cells. Another is autoimmunity. I don't pretend to understand this. I don't know if anyone does. This is sort of a catch-all term or a black box, but when the immune system reacts against self proteins. And then you have all of these other factors: stress, lack of exercise, lack of sleep, neurotoxins like heavy metals and pesticides and other agrochemicals, insufficient anti-inflammatory foods, and so forth. So there are many things that can cause inflammation. One that I want to talk about is the gut-brain axis. So it was said by Hippocrates, the father of modern medicine, that death begins in the gut. And traditional Chinese medicine and Ayurvedic text, writing some thousands of years prior to that, actually said, interestingly, we agree with Hippocrates on this one, I'm paraphrasing, and that they all argue that the strength and quality of digestion and the gastrointestinal system is critical to the aging process. Because, you know, the GI tract is actually outside of the body, technically. It's just a long tube, kind of like the internet. It's just a series of tubes, right? So, so that's a tissue that we really need to protect, and it's responsible for assimilation of nutrients. And when that process breaks down, we become deficient. Even if we're eating enough of some of these nutrients, if your gut is not absorbing them, that's a problem. And actually, in my own work in the lab, previously did a Fulbright fellowship in a lab working on telomere biology in zebrafish. And we found that the first tissue to decline when you knock out telomerase in the zebrafish is actually the gut. And that makes sense because the gut is the most proliferative tissue, or one of the most proliferative. Hematopoietic stem cells are also highly proliferative. And interestingly, when you knock out telomerase, that's really bad for the HSCs in the bone marrow too, as a side note. Another great thinker who called attention to the gut-brain axis, or the microbiome before it was cool, fashionable, was Elie Meshnikov. He won a Nobel Prize for his discovery of macrophages and was co-awarded with Paul Ehrlich. He was one of the first biogerontologists as well. So he studied population of long-lived Ukrainians, they attributed their longevity to fermented foods and yogurt. And so he was an avid yogurt eater for the rest of his days. And he did live a pretty long time, I believe. So he studied the gut microbiome before it was cool, and he knew a thing or two about the immune system. So maybe we should follow up on what he said. So what are the physiological mechanisms underlying the link between gut permeability and brain problems? Well, here's one. You have a bad diet and lifestyle. So you have high sugar, low fiber, lack of exercise. Maybe you're eating a lot of molecules that induce gut barrier permeability, such as gluten found in wheat. You can believe the hype. Gluten, even if you don't have celiac disease, in some proportion of the population, they have non-celiac gluten sensitivity. Gluten is a protein that punches a hole in your gut barrier in the one cell layer thick epithelium. It binds a protein called zonulin, pops open the tight junction, and then stuff gets across that membrane that shouldn't get there. So it's leaky gut syndrome. And you can look into the work of Alessio Fasano, who was at Harvard, who detailed the molecular basis for celiac disease. And he also says that gluten punches a hole in the membranes of pretty much everybody, even if you don't have celiac. Whether you get symptoms depends on your immune system and some of the MHC polymorphisms that you possess. So anyway, there's a lot of stuff that can cause gut barrier permeability. This can also cause dysbiosis, so the growth of bad microbes that shouldn't be there, like Candida albicans if you're eating a lot of sugar. This causes local inflammation, and that causes peripheral inflammation. If you have leaky gut, you have molecules such as food peptides from proteins not fully digested to their amino acid constituents that get across that membrane, and then they're recognized by dendritic and other immune cells. That acts almost like a vaccine. So when you have a leaky gut and you eat foods that are immunogenic at all, you become more responsive to them. And so you're basically vaccinating yourself against your own food. And that creates de novo food allergies and also autoimmunity if the peptide sequence of what you're eating resembles the peptide sequence of a self-protein elsewhere in your body. So there's a molecule lipopolysaccharide. It's a PAMP that is detected by the intestinal epithelial cells. This activates our favorite protein, NFKB, which is a master regulator of inflammation. That results in secretion of pro-inflammatory cytokines like IL-1 beta. IL-1 beta, interestingly, increases both gut barrier permeability and also permeability of the brain because it circulates, right? It's a circulatory factor. So IL-1 beta and probably other cytokines are actually linking the permeability of your gut to the permeability of your blood-brain barrier. So the microbes and LPS and other PAMPs get into the brain, induce cellular senescence, neuroinflammation, and other bad stuff. If that persists, you get T cell infiltration, and that's really bad. And we'll get into that, so show some data on that. You get cytotoxic T cells coming in, and those T cells can actually poke other cells and tell them to apoptose. And so that is what we seek to avoid. If that happens, it results in neurodegeneration. So if the microglia can't do the job, then the big guns come in. These are the T cells, infiltrate the brain, and they start killing off neurons, as you can see in this confocal image here. So the dogma for a long time was that the blood-brain barrier keeps T cells and peripheral immune cells out of the brain. That they also believed that the brain was sterile, that there were no microbes present. Even in healthy people, we are finding microbes in the brain, especially if you are not healthy and you have barrier permeability, then we're seeing a lot more microbes in the brain. So one of the exceptions, one of the results here in 2019 showed that T cells, there's a paper I'll show you, showed that T cells repress neurogenesis found in the aged hippocampus and the subventricular zone during memory loss. So this was a very cool paper from the lab of Anne Brunet, who is also a major force in the aging field. And she and others showed that T cells infiltrate the brain and specifically get into the neurogenic niches where new neurons are created. So the T cells get in the mouse brains, they go to the niches, and they secrete interferon gamma, which is sort of an antiviral response cytokine that is sufficient to inhibit neurogenesis both ex vivo in dishes and in vivo and compromise cognitive function. So as a brief anatomy lesson, at least in the rat brain, you have several areas of neurogenesis. In the olfactory bulb, because these animals have a really keen sense of smell, in the subventricular zone, and also in the dentate gyrus of the hippocampus. And between the olfactory bulb and the subventricular zone, from the subventricular zone, you have the rostral migratory stream. We don't have that in humans, or very little. And it's pictured here, the subventricular zone, right outside the ventricles. And one of the reasons there's neurogenesis right next to the ventricles is because the ventricles contain CSF, cerebrospinal fluid, that bathe these neurons in signaling factors, such as growth factors and neurotrophins that tell them to proliferate. So in that paper, Brunemeier and others found that in young animals, there is not very much infiltration of these CD3 positive T cells in the, say, subventricular zone. But in old animals, there are more. Say you can see about 4 of these cells in this section here, and it's quantified there. So you're getting more T cell infiltration in aged brain. And also those T cells rife in the aged brain, they're actually releasing interferon gamma, and they're expressing, as you can see, BST2, this interferon gamma responsive gene. It's quite a substantial effect size. Also in post-mortem human tissue samples, you see more T cell infiltration. So in the young human, you see few CD3 and CD8 positive T cells, but in the old, you see more, and that is quantified here. Switching gears a bit, one of the most intriguing interventions in the aging space that's gotten a lot of press attention because it's kind of vampire-y and captures the public attention is heterochronic parabiosis. So you take the circulatory systems of an old and a young mouse and you link them such that they share blood. The old mouse gets younger, the young mouse gets older. The young mouse gets more old than the old mouse gets young, which indicates that there's actually more bad stuff in the old blood than good stuff in the young. A lot of this work was pioneered in the Bay Area at these institutions, and some of it is being taken forward by this Spanish blood products company, Grifols, now. And they recently announced some compelling data, not that recently, over a year ago, I guess, data finding in Alzheimer's disease that if you actually do plasmapheresis, you scrub out the blood and you just replace it with saline plus albumin, a blood component, it actually works quite effectively. One of the largest effect sizes we've seen, even in moderate dementia patients. So not most Alzheimer's trials are in mild patients. These are actually in moderate patients. And we're seeing a pretty decent spread in the data there. So it's a 60% reduction in progression, even in moderate AD. This comes out of the work of primarily Irina Conboy and Michael Conboy's labs, as well as Tom Rando and Tony Vizcorre's lab. Plasmapheresis, you scrub the blood. It's kind of like bloodletting, which has been done for millennia. There may have been something to it, but you replace the albumin. Sadly, this is not one single profitable drug, a pill you can pop, or an antibody you can inject. So pharma hasn't really paid a lot of attention to it because it's not scalable, it's not really patentable, but thankfully Grifols is. And by the way, I do own stock in Grifols, although not very much. Okay, so coming on to the amyloid hypothesis in Alzheimer's, I have a very substantial bee in my bonnet about the amyloid hypothesis, and hopefully you'll see it's for good reason, because this flawed belief, this dogma forced upon the field for about 30 years has kept new therapies away from dementia patients, including a member of my family, for 30 years, right? So let's look at the sad reality. In this time period, there were only 2 mechanisms of action drugs approved versus all of these failures. So pretty much everything has failed. In Alzheimer's, there are about 250 trials that got into phase 2 or phase 3 that failed, and only 2 drugs that were approved. And by the way, these drugs, donepezil and memantine, they barely work. They slow the rate of progression a little bit, but they have bad side effects. So we cannot declare victory on the basis of those 2 drugs. So why would this happen? Why would there be such a consistent rate of failure in Alzheimer's versus other clinical indications? One, the paradigm is wrong. Most of these drugs that have been tested are reliant on the amyloid hypothesis, and we'll define what that is in a minute for those that are not aware. Another is that we're simply intervening too late. By the time memory becomes a real problem for which you seek treatment, the pathology is too far gone. You're already starting to lose neurons at that point. And they're also not stratifying patients based on the Alzheimer's subtype. So AD is not a single disease. There are multiple causes It's just lumped together as one thing, presenting as dementia and cognitive impairment. We'll come to that later. So sporadic AD is not a single disease with a single cause. There are genetic forms of AD, but they're like 1% of the actual population of people with AD. Lumping them together, they're overlapping pathologies based on just a memory phenotype. There's no biochemical basis. People say, oh, it's A beta or tau presence in the brain, but newsflash, most clinicians are not checking A beta and tau levels in the brain of patients. And when they do check, they find that there's a pretty weak correlation between cognitive decline and these aggregates. So it's a form of dementia diagnosed with memory tasks. There are multiple subtypes based on the work of people like Dale Bredesen, who I'll talk about. One is a type 3 diabetes, CNS insulin resistance, but can also result from mitochondrial damage, glucose hypometabolism, you can't burn fuel, and then IDE, insulin degrading enzymes. So when your insulin level is high, IDE is busy degrading insulin, and it cannot degrade this other junk that accumulates in cells, including amyloid beta and other proteins. Neuroinflammation we talked about can be caused by undiagnosed traumatic brain injury, poor sleep apnea is a big one, failure of the lymphatic system to clear things out. Pathogens, blood-brain barrier leakiness, and ApoE44 is a major risk factor for Alzheimer's, and it actually enhances the inflammatory response. Can get into that in the Q&A if you want. Also, exposure to toxins that are not commonly checked for. Mold toxins like aflatoxin, a super ultra potent DNA damaging agent. Metals like mercury from dental amalgams, pesticides in the food, industrial chemicals in the food and water, etc. Loss of trophic support. So we have these pro-growth and pro-cognitive factors like brain-derived neurotrophic factor and nerve growth factor, and also neuroactive steroids, pregnenolone and estrogens are very important. And this is one reason why menopause is quite commonly inducing cognitive decline, and why women have about twice the risk of Alzheimer's as compared to men, unfortunately. That you actually have vascular dementia, not whatever Alzheimer's dementia actually is. And this can be caused by a lack of nitric oxide, cyclic GMP, and Krebs signaling. Krebs is the master regulator of memory and learning, and they're working with a group with a drug that enhances this activity. And just as a side note about nitric oxide, one of the reasons you want to breathe through your nose is because there are cells in the nasal cavities that produce nitric oxide. Nitric oxide is a very important neurotransmitter, but it's also a vasodilator, and also a antimicrobial agent that is important for lowering your blood pressure and increasing perfusion, blood and nutrient perfusion into tissues, which declines with age. So always breathe through your nose whenever possible. And there's certainly other subtypes, and they're all interlinked. You can have multiple of these problems. A lot of this is based on the work of Dr. Dale Bredesen, who's a neurologist and the founder of the Buck Institute for Research on Aging, which is the largest private aging research institute in the world, where I previously worked. And he has set up a number of clinics and this protocol that has been used all over the world now. And it is the most effective intervention for patients with Alzheimer's disease, better than anything known. And we'll talk about that in a minute. So Alzheimer's, it turns out, is preventable. The dogma in the field, amazingly, is that it's not preventable with any kind of intervention. This struck me as absurd because poor health accelerates aging, and aging is the main risk factor for Alzheimer's. So why would it not be the case that if you slow aging, you can actually slow your Alzheimer's risk? Furthermore, Alzheimer's has increased out of proportion with longevity gains in the population, so there are modifiable risk factors. So if you slow aging, hopefully soon with drugs, in addition to diet and lifestyle, you should be able to slow Alzheimer's, and check out these references for more evidence. So the amyloid hypothesis, people are now saying it's failed. Correlation is not causation, and the correlation wasn't very strong to begin with. So blaming A beta for Alzheimer's is like blaming the firefighters for the fire. Yes, they often, but not always, coincide in time, but maybe the firefighters and the A beta are actually trying to solve the problem. They're there to try to help out. And it turns out that A beta, like most proteins in the body, didn't evolve to cause us problems. Every protein and gene evolved to serve a function that promotes health, or at least promotes reproductive fitness at the very least. So these drugs have failed because they're mostly based on the Alzheimer's hypothesis. 80% of the drugs have failed. Many other drugs have been tau focused, and I will submit that tau is not the cause of sporadic AD either. Wasted billions of dollars over 30 years. I strongly recommend reading this article from Stat News in 2019, how prominent academics suppressed alternative theories and counter evidence against the amyloid hypothesis, including Dennis Selko at Harvard. Definitely read this because it's a great masterclass in how academics can enforce dogma even when the evidence is not in their favor, how there's a lot of bias. Scientists are not paragons of rationality and enlightenment. They are, you know, opinionated and political and corruptible and prone to error as many others. And Max Planck, the great physicist, once said that science advances one funeral at a time. So maybe A beta is not the bad guy. Maybe it's actually good. Maybe it's, by one theory, functioning as an antimicrobial peptide. So it's in some dementia brains, but not all. Many people have high A beta plaques, but no dementia. Correlation is weak. Drugs that remove A beta from the brain effectively, like aducanumab, don't do anything really for cognitive enhancement, although the FDA is considering approving it just for political gain. So what if A beta is there to help? And maybe it's an antimicrobial peptide. So there's a process called opsonization. You can do it with antibodies where there's a pathogen or some other problematic molecule like a protein aggregate. It gets surrounded by antibodies or by A beta, and that attracts immune cells such as NK cells to come eliminate it, and that solves the problem. So it's opsonization, it's a neutralization mechanism. Some work from Rudy Tanzi's lab and others shows that A beta peptide actually ensnares and encapsulates and you know, holds back microbes, including salmonella. Here you can see these fibrils capturing and trying to neutralize salmonella, as well as agglutinating candida. If you eat too much sugar, candida accumulates as well. So A beta is an ancient protein. It's found, you know, for hundreds of millions of years in very distant genomes of various organisms. So it's obviously doing something important. It did not evolve just to cause Alzheimer's, right? It's something going wrong. So these microbes, where are they coming from? Well, one theory is some come from the mouth, gingivitis causing microbes. There's a company that was a very successful IPO a couple of years ago, Cortexime, developing small molecule inhibitors of a gingipane protease produced by one of these microbes. I actually don't think that will work, but it's gotten a lot of attention as a new approach to Alzheimer's. But another good one is the gut, for the reasons that I mentioned previously. And why the gut? Well, it is the interface with the immune system in the outside world to a large extent, and it's very fragile. It's only one cell layer thick. The intestinal epithelium is only one cell layer thick. Fortunately, it's very proliferative, so you can replace damage relatively quickly. But if there is breakage and interruption in that one cell layer thick, you've got a problem. So what can we do about it? Well, this is a kind of surprisingly underdeveloped area of drug discovery. I mean, for the last several years, I've been scouring the world for therapeutic approaches, and I've rarely come across approaches for restoring and enhancing gut barrier integrity. But one that I've come across recently is this protein called intestinal alkaline phosphatase, IAP, that comes out of the lab of Richard Hoden, who's the head of gastroenterology at Mass General and Harvard Medical School. He's a lovely guy. We have come up with ideas on how to advance this molecule in the clinic and it's been partnered with an existing company who looks to be interested in advancing this protein. Anyway, so why is this of interest? It's backwards. All right, it's produced in the gut. Its levels decline with age. It's produced in mother's milk to train the immune system. It modulates the gut microbiome. It enhances gut barrier function, proliferation. It's already worked in a small clinical trial in ulcerative colitis previously. And most interesting for me as being a geroscientist is it extends mouse lifespan by 30% median with no apparent side effects. This is the world record for lifespan extension. And other drugs that do extend lifespan like rapamycin, metformin, et cetera, they do have side effects. So this is a very interesting approach, and I'm working on trying to get that in the clinic ASAP. Another interesting area is neurotropic viruses. I'm not an expert in this, but this is a hot area and it's not new. So to my knowledge, there are no beneficial viruses. Some microbes like bacteria are symbiotic, but to my knowledge, all viruses are either commensal, they don't do anything bad, or they're actively pathogenic. Some viruses infect the nervous system selectively, the herpes group, picornaviruses, rabies, measles, mumps, rubella, and even HIV has tropism into the brain. Another one that is relatively understudied are these endogenous retroviruses or transposons. These are strongly implicated in aging and brain aging, and we can come to that later. One of the pioneers in this field who had been criticized for many years until it was proven that, looks like she's right, is Dr. Ruth Idzaki. She's currently or was recently a visiting fellow at Oxford, so you can go visit her there. She found HSV in 80 patient brains a long time ago. Many studies have found a link, and you can follow up on this research and some of these reviews by Dr. Idzaki. So this is herpes simplex virus. Almost everybody has it, and almost everybody has a lot of different viruses, many viruses we have never even defined, but our immune system is strong enough to keep them in check. Just like usually our immune system is strong enough to keep cancer in check. So the question shouldn't be what virus is causing the disease. It should be what is causing the failure of your immune system to hold at bay these pathogens. So it is a little bit of a controversial field in dementia. Some studies have found a link, others haven't. I tend to believe that there is a link. It may be a specific subtype. In fact, I'm quite sure it's a specific subtype of Alzheimer's that is caused by pathogens, including HSV. And, it could just be that the HSV presence is a secondary side effect of the failing blood-brain barrier immune system. So the HSV is not the primary cause, but it's just sort of a readout on the integrity of the blood-brain barrier. Nonetheless, there was a very large study finding that if you give patients who are taking anti-herpes simplex virus drugs, small molecules, they have a 2.5x reduced risk of Alzheimer's disease. It actually reduces your risk of Alzheimer's back to baseline, which is quite compelling. So it's worth investigating further. As an interesting development, I guess I'll throw this out for my advisor at Oxford, who is an expert in DNA damage repair. There's actually a link between brain aging and memory and learning and DNA damage. And it's not what you would expect, and it surprised me, which is we require DNA double-strand breaks in order for memory and learning to occur. DNA double-strand breaks are usually a pretty big deal in the cell, pretty bad, cause apoptosis. CRISPR causes DSBs. That's a drawback with CRISPR and why the nicases are more interesting. But several labs have now found, including the Li Hui-Sai lab at MIT, that these breaks are actually critical for memory and learning. They're a switch that turns on early response genes and synaptic plasticity and requires topoisomerase 2b beta. And in neurodegeneration, these DSBs are not repaired. They accumulate and they cause inflammation and apoptosis. So I mentioned these subtypes of Alzheimer's. Dale Bredesen is one of the leaders in this field. He was trained at Duke, UCSF, and Caltech, and he's a professor at UCLA and was at the Buck. And he has come out with his book, The End of Alzheimer's, that details these hundreds and hundreds of patients that he and others have actually helped better than any other intervention known with Alzheimer's. And it's not a single drug. Unfortunately, it's not that easy. It's a pretty radical change to diet, lifestyle, and taking a lot of supplements, including B vitamins that are missing, and clearing out toxins from the brain, including mercury and other such insults to the brain. So, so definitely check out this work, and I think it's going to get a lot of attention soon enough. And we covered some of these mechanisms previously. Brain energy metabolism is a really appealing area. And so here are some of the drugs that are in development in brain disease that actually act upon brain energy metabolism. You can look into these at your leisure. Not described here specifically are enhancers of mitophagy as well as mitochondrial biogenesis. So clearing out the non-functional mitochondria and replacing them with functional mitochondria. So I can wrap it up here. So to conclude, I just want to throw out some more ideas to you guys for further investigation that I think are worth exploring. So metabolic syndrome is the big one, I think, particularly in America and the West, where our diet is so bad, laden with carbohydrates and high glycemic index foods. We have insulin resistance in the brain, causes problems with mitochondria and bioenergetics. It's also linked to cancer metabolism. Nutrition, so what is the optimal intake of micronutrients beyond the recommended daily allowance, which is not suited for optimality. That is just what you need to not get an acute disease. How much of these nutrients do we need for optimal health? And also the avoidance of dietary toxins like gluten that I mentioned, but also lectins and other plant foods. Infectious agents we've talked about already. Enhancing phagocytosis and debris clearance. There's a fair amount of work going on at Oxford actually on this in the Alzheimer's Research UK Center there. The lymphatic system in sleep. So we actually have the lymphatic system that circulates blood cells and clears debris. Well, it turns out the brain has it as well. This was discovered relatively recently. This is one reason why sleep is so important. There are many reasons, but one of them is because it allows the lymphatic system to clear out the junk and debris from the brain. The mechanism of action of ApoE, this is a major risk factor in aging, including dementia, but also cardiovascular disease, figuring out how exactly that causes disease. The role of nitric oxide and Krebs signaling in the brain, this is something I'm working on right now. And also enhancers of neuroplasticity. So there are nootropics, low toxicity cognitive enhancers out there, as well as the psychedelics, which are gaining in popularity. You may have heard in the UK, Compass Pathway is a company working on psilocybin, as well as Atai working on a number of other psychedelics that enhance memory and learning. Sadly, 1 in 8 people will get Alzheimer's disease, and it could be you. So I hope this is an invitation to you to get cracking on the research and try these new approaches that are not what has been orthodox and what has already been tried to, almost to excess. We need to try new approaches. And I know in academia that's hard because there's this sort of consensus-based incrementalism, and this is why we need fresh approaches that are sort of high risk. So for those of you who are interested in the longevity biotech field, I'll give a plug to some of the more prominent groups in that space. The ones above here are the ones that I have worked with: Apollo Ventures, Samsara Therapeutics, and Cambrian Biopharma. Samsara is hiring biochemists in the Oxford area right now, so get in touch, as well as Cambrian hiring people in New York, but also remotely. And these are some of the other groups in the aging space doing very interesting stuff. And you can see here this link to a bunch of other biotech companies in the aging space, with the caveat that many of those companies on that list are not really truly in the aging geroscience space, but are kind of tangentially related, but nonetheless, do check them out. So I will conclude with that. And on a bright note, there's never been a hotter time in biotech and for academic spinouts. So we're sort of in the golden age of doing it. Things may be a little bubblicious right now, but I think we are, reaching sort of a new plateau as biology becomes more like an engineering discipline, and we're able to advance all of these different modalities. For 100 years, it was just small molecules. Now we have antibodies and biologics and peptides and gene and cell therapy and, siRNA and all the rest, right? So drug discovery is really becoming a much more developed discipline. And it is often said that this will be the century of biology. The last century was the century of physics and chemistry and the engineering of those disciplines. Now I think we're coming into the century of biology. So on that note, thank you everybody for listening, and I'm happy to open it up to questions.
Brilliant. Thank you, Sebastian. That was incredible. Absolutely terrified with my insomnia-wrapped brain for the future.
But I haven't thought about it.
We'll see.
Yeah, man.
So I'm going to kick off the questions. I've got one for myself, actually.
Sure.
So I'm going to go right back to the beginning where you were talking about proteostasis and synapse integrity. And I wanted to see if there was a link between the actual sort of cell autonomous proteostasis and its regulation in sort of those synapse nodules, if that makes sense.
I'm not sure I understand the question. Could you rephrase it?
So you were talking about how immune cells prune the synapses, and that's sort of a mechanism to the brain aging. But also within the neurons themselves, and is sort of neuron proteostasis linked to sort of synapse degeneration and loss?
Yes, absolutely. So a lot of the proteins synthesized in the ER actually have to be trafficked into the synapse, either, basically into the dendritic spines. Sometimes they're synthesized there. And if there's a problem with that process, you get die-off of the dendrites. So if you have a failure of the integrated stress response, which I didn't get into here, but I previously launched a company out of the Scripps Research Institute working on a mechanism related to the integrated stress response, which is becoming hot in the aging space, and its focus on neurodegeneration. So the integrated stress response happens when there are various types of stress, but one of them is proteostatic stress. And the cell basically stops everything and it slows down translation. And it basically allows time for the cell to keep up with that proteostatic stress. And it doesn't slow translation across the board. It slows translation of certain very highly translated genes, but not others that are involved in proteostatic repair and recovery, like chaperones and so forth. So the way to think about it is you have an assembly line, and the assembly line has widgets passing along it, things that need to be operated on by a robot arm or something. And with aging, the pace of that assembly line does decline. The translation rate does decline a bit, but it does not decline as much as the decline or decrement in proteostasis and repair and proper folding and degradation, turn over those proteins. So if you slow the pace of translation, that allows time for the proteostatic machinery to catch up and fix the problem. So it's absolutely the case that there is a cell autonomous, there are cases of cell autonomous proteostatic stress causing loss of synapses.
Perfect, thank you. So in the chat, there's a question from Robert who asks, is there any known relationships between autophagy and apoptosis?
Yes, there is a very strong relationship. And actually, I would direct you to the work of Guido Kromer in Paris, who is a very clever guy who's been working on this for years. And it was often whispered that he would get a Nobel Prize for his work on apoptosis. But then there was an apoptosis Nobel awarded. And so he switched to autophagy, probably to still be in the running for the Nobel Prize. And yet, anyway, so so he's focused on the interface of apoptosis and autophagy. And put simply, autophagy is the first resort. If you can solve the problem with autophagy, you don't need to do apoptosis. But if autophagy fails, cells are going to apoptose as a last resort. And there are, great review articles from Kromer and others about that link between apoptosis and autophagy. And there's also an interesting link to cancer as well, which is some but not all tumor cells turn on autophagy as a way to prevent apoptosis and enhance their cellular fitness and resilience, also to resist chemotherapy. So we can actually learn from cancer cells, just like we can learn from the germline and the embryonic stem cells on their mechanisms of resilience and immortality, because cancer cells are immortal. So they've evolved these mechanisms to live forever. And so we can actually harness that learning for the rest of the soma, the other non-transformed cells in the body. I should note that it does not follow that enhancing autophagy is going to cause cancer. In fact, we see the opposite in animals. If you reduce autophagy, it causes a lot of damage that makes cancer more likely. And autophagy is critical for immune function, as you can ask Katja Simon there at the Kennedy Institute in Oxford, who is a world leader on autophagy in the immune system. So I'll leave it at that.
Susanna asks, does a ketogenic diet mimic the effects of fasting on autophagy?
Yeah, so it does mimic some of the benefits of fasting, but really it doesn't quite go all the way in autophagy, because with a ketogenic diet, you're still consuming a lot of calories. And if you have a lot of calories, autophagy doesn't turn on very strongly. That said, the strongest inhibitors of autophagy are the most insulinogenic foods, macromolecules, which are carbohydrates primarily, but also proteins. And proteins, amino acids, particularly leucine, activate mTOR, and mTOR represses autophagy. So you do get a slight increase in autophagy, autophagic flux, with a ketogenic diet. And there are other really good things that a ketogenic diet or even exogenous ketones can do. I take exogenous ketones in part because ketones like beta-hydroxybutyrate act as HDAC inhibitors, histone deacetylase inhibitors. And that causes the de-repression of a lot of genes that are involved in memory and learning. So there are obviously merits to the ketogenic diet, especially in diseases like cancer and glioblastoma. but it doesn't turn on autophagy as intensely as, say, a water fast or just a relatively low calorie diet does. But it's really hard, and I've tried to do full bore caloric restriction. I do intermittent fasting, but to really get your caloric intake down substantially by 33% or more, it takes a lot of willpower that I personally don't possess. And so wouldn't it be great if we could take a pill that mimics not only the benefits of exercise, but also the benefits of fasting, particularly autophagy? So I and other groups are currently working on that. And I'm pleased to say within Samsara and these other groups, we have made a lot of progress. I mean, we have molecules that safely and potently induce autophagy and drug-like concentrations with good pharmacokinetics. So this is, a perfectly viable approach. And we've already seen with rapamycin that enhancing autophagy does extend lifespan. So anyway, you should try the ketogenic diet, but it doesn't boost autophagy as high as I would like to see.
Back to Robert, who says, what does it take roughly to recruit an SAB like the one at Samsara? And also, what at Samsara do you work on autophagy resistant substances like lipofuscin?
Yes, good questions. Recruiting an SAB, as I've done a handful of times for biotech startups, is not as difficult as you would expect if your science is really rock solid. So, the team at Samsara is, some of the most prominent in the field. And, it was funny because when I was going out to launch a new company at Apollo, I basically was given the sort of a blank check proposal to just survey the aging literature and find an area where we should start a company where we don't already have a company. So I interviewed about 100 PIs, professors all over the place, and ultimately met Guido Kromer, Frank Matteo, and they were actually kind of pitching me to start this company because they had been working on this for a while and it was getting to a level of maturity that it was ready for a spin out. So pulling together the team was actually relatively easy in that case. But just, understanding who's at the top of their field, you become, not quite an expert, but know a thing or two about the space. You can interface with them and you can recruit an SAB. It's not too difficult. As for lipofuscin, we do see with autophagy enhancement, pharmacological and genetic, that you do degrade lipofuscin. There are some molecules present within lipofuscin that cannot be degraded by known mammalian enzymes. One is A2E, which is a retinoid metabolite, and groups like the SENS Foundation, a very important funder in the aging space and sort of philanthropic organization, have been working on mechanisms to degrade these undegradable molecules. Some of them are oxidized cholesterol. Some of them are like this pigment, photoreceptor pigment A2E. And one way you can do that is you can actually engineer enzymes from microbes. This is called medical bioremediation. I wrote a review article on this years ago that can degrade those undegradable molecules that accumulate in humans. We just need the lock and key. We need a new key that can actually enzymatically catabolize those molecules. So we do see a reduction in lipofuscin, but there are still some molecules in lipofuscin that even autophagy cannot degrade. That said, autophagy and lysosomal exocytosis can actually get rid of junk that cannot be degraded by our presently available enzymes. So molecules, say like A2E, that would accumulate, we can actually spit it out of the cell with lysosomal exocytosis, and then hopefully it gets into circulation and then it's eliminated by the kidneys or the liver. So we don't actually need to degrade it. We can actually just eliminate it. So hopefully that answers your question.
Perfect. Question from Marco. Really nice talk. Could you comment on the fact that in a few papers, when you treat cells with a synthetic amyloid beta oligomer, they actually act as toxins and decrease the viability of these cells?
Yeah, of course. Well, first of all, the dose makes the poison, right? I can treat a neuron with many different molecules and it'll be toxic at a certain dose. But A beta is actually pretty potently toxic. And it's not to say that A beta is not bad for neurons. Indeed, it is. If you read Dale Bredesen's book, The End of Alzheimer's, he goes through the molecular basis of the degradation of amyloid precursor protein, APP, into two different pathways, one of which results in the formation of 42 amino acid A beta peptide, and the other results in like alpha CST and these pro-survival peptides. So APP is actually a sensor for various stressors, kind of like mTOR is. It's a central regulator of various nutrient availability and cellular conditions. Amyloid precursor protein, this ancient gene, is actually sticking out of the cell surface, and it is sensing all of these different inputs. And if there is a certain level of stress, then it will be cleaved into certain length of peptides that are what Bredesen calls synaptoclastic. So you have synaptoblastic and synaptoclastic signals, building and degrading signals, just as you have in the bone, osteoblastic and opteoclastic signals. And the osteoblastic signals build bone. And so just the same, APP is acting as a central regulator. And when there is that stressor, it results in A beta production. And A beta tells the cells to actually shrivel up. And in addition, I mean, it would make sense because proteins rarely evolve for one function only. They usually multitask. That's kind of the beauty and also the difficulty of evolution. Nature is parsimonious with its parts, so it uses them for multiple functions. And so A beta could be opsonizing microbes. It could also be acting as a pro-inflammatory stimulus to reduce cognitive function in order to do that sort of rest and digest, chill out signal to try to solve the problem. But it also is synaptoclastic, causes the shriveling up of synapses. So, but those kind of models, those really simplistic models where, you know, you have some kind of cell stressor in a dish, really have led us astray over the years. It's better to look in vivo and look at wild type aging if at all possible. These animal models, like these Mouseheimer's models, we can cure Mouseheimer's in many different animals with many different interventions, and it hasn't worked. So maybe it's time to look at these different models. And one model that I particularly like is just wild type mouse aging, or these accelerated aging models, SAMP8 or progeric mice, because you have something that really looks like wild type human aging. And if you can slow that, you're probably going to be able to slow real human aging. And it's not a contrived fake animal model disease where the animals are expressing, you know, 10,000 times the level of A beta they would ordinarily express. It's just regular aging. So anyway, I'll leave it there.
I think that's it for questions in the chat. Sebastian has agreed that if you do have any questions that you could reach out to him via LinkedIn, if that's still okay, Sebastian?
Yep.
Yeah, perfect. And yeah, thank you very much everyone for joining. Thank you, Sebastian, for giving a talk today. For the second time. It's great to have you.
Absolutely.